All right, so welcome to this masterclass from Cambridge Leadership College. My name is Warwick Bailey. I'm one of the Z coaches here at the college. And in this masterclass, we're looking at AS level biology. And this is a structured paper from 2013. So in these structured papers, paper 2, the paper just runs for just over an hour and 15 minutes. And there's just quite a lot of short answer questions, but a few where you're required to give much more in-depth answers. And I noticed actually from both the mark scheme and particularly the examiner's report actually gives a lot of depth. There's actually quite a lot that you can read from the examiner's report. So my plan for this masterclass actually is not to go through every single question, but I'm going to focus on questions 2 and question 5. Okay, they're both quite high mark questions, but rather than just trying to repeat what is said in the examiner's report, I think I'll try and do a deep dive into questions 2 and 5 and try and go a bit deeper into some of those questions and just kind of go beyond what the question's saying to take a deeper view at some of the issues that are raised in the question and in the biology and biochemistry behind that question as well. All right, so I'm going to skip through question 1 and then spend a lot of time really on question 2. Okay, so there's a number of, I suppose they'd be called inorganic ions. And there's a question here about where these ions are particularly used in different areas of biology. And so the first one is a component of polynucleotides. Let me see if I can get that into red so that's a bit clearer. Okay, so I don't think that's a term that's used that often. Clearly, this is a polymer of nucleotides, polynucleotides. And you're much more familiar with calling these polynucleotides either deoxynucleic acid or ribonucleic acid. So these are polymers of nucleotides. And so in terms of getting into a bit more depth behind this, the nucleotide is composed of 3 components that you need to know about. One is a 5-membered ring sugar, that's a pentose sugar. Then phosphate and the bases that make up the nucleic acid. So these polynucleotides, they're polymers of nucleotides. The nucleotides themselves are built from 3 components. We'll look at those in a bit more depth in a moment. And you're much more familiar with calling those polynucleotides nucleic acids. Okay, so that's the same thing. A polynucleotide is the same as a nucleic acid. So let's just look at these 3 components in a bit more depth. I find some space down here. So you've got the sugar, the phosphate, and the base building the nucleotide, which is building the polynucleotide. So the sugar, you need to know, is typically drawn as a cyclic form, but it's a bit easier to understand it if you draw it as its open chain form. And the sugar has this functional group at the top, and if you're doing A-level chemistry, you'll recognize that immediately as an aldehyde group. And then you come down to just a CH2 group there, because this is a deoxy ribose. And then you've got OH groups. You've got another OH group. And then you've got CH2OH. Okay, so if you just count up there, you've got 1, 2, 3, 4, 5. So that is sometimes described as a pentose sugar because you've got 5 carbons in the chain there. Another descriptor of it is because you've got this aldehyde group up here, that's an aldose sugar. And because these aldehyde groups can be oxidized, then the sugar is called a reducing sugar. So you can see there's quite a lot of overloading in the ways that you can describe this molecule. It's a pentose, it's an aldose, it's a reducing sugar. Just on its own, it's a monomer, so you can call that a monosaccharide. And because at that point there on carbon number 2, you can see we're missing one of the familiar alcohol groups. It's a deoxy ribose, okay, because we're missing one of the alcohol groups there. So that's its open chain form, and a ring form of this molecule can form because a bond can go from number 4 all the way around to number 1 there. And so it's much more familiar to have that as this ring form, like this. And so that bond there between 1 and 4 is between 1, 2, 3, 4. You can see the 1,4 bond there. And then from the 4 position, you've got this CH2OH. And when that bond formed there, a new alcohol group was created. In that arrangement, this is an alpha deoxy ribose. Okay, and at the number 2 position, there's no alcohol groups. The number 3 position, there is. And the alcohol group that was at the number 4 position is now part of this ring system here. Okay, so that's deoxyribose, and in solution, the cyclic form is the one that's predominant, and then this open chain form is only there in small quantities, but it's sufficiently there so that this aldehyde group can be oxidized, and that's why this is called a reducing sugar. So that's the sugar, or the pentose sugar component, the nucleotide. The next component is the phosphate. Now you can see here, if we take phosphate, which is, if you know your chemistry, looks like that. And it's going to be a condensation reaction between that alcohol group there and this phosphate here. So what happens is you get this coming down to the phosphate. Okay. And then there's another condensation reaction which joins on to another one of these deoxyribose monomers, and it's going to come in at carbon number 5 there. Can you see like that? So this is the next pentose sugar, and we've got that ring system again, and that's come in there. We've joined, that's for number 3, isn't it? That's at number 5, and then the pattern continues because that's number 4, 3, 2, 1, and then the pattern continues as you go down here. So that's going to join down to another, if I can come down here, to the next phosphate. I'm running into the next question, but I'm running out of room a bit here. Okay, and then that joins on to the next 5 carbon, and so the pattern continues down there. All right, so you can see here that as you build up this backbone, it's called, of sugar and phosphate groups, that backbone is being built up through the number 5 position and the number 3 position. And so one way to describe this is to say that you've got a chain which is running in the 5-3 direction. Okay. Now the Next part of the polynucleotide then is to add in the bases. So if I take out some of the things that I drew before, and then the bases are going to be added in at this one position here. So there's another reaction where instead of having that alcohol group there, at each of these one positions, we're going to have a base added. And you can see I'm starting to build up this polynucleotide. I've just got 3 bases there. These DNA molecules can stretch to thousands of these bases along here. So this is just one strand of a polynucleotide. For DNA, you have 2 strands, don't you? So there's one strand here which is going in the 5'3" direction, and there's another strand which is going in the 3'5" direction. So these 2 strands are called anti-parallel, and you could draw out the other strand over here. And those 2 strands are held in their double helix because you form hydrogen bonding between the bases on one strand and the bases on the other strand. And so those bases form complementary base pairs. And one of the features of the complementary base pairing is that each of these bases here is either going to be based on a purine base or pyrimidine base. So the purine base has you don't have to know these structures, but you have to know the base pairing. So the purine base has 2 ring systems. The pyrimidine base has a single ring system. And when you have this complementary base pairing, there's only enough room between these 2 strands for a purine and a pyrimidine. You can't fit 2 purines in there. And so whatever the base is on this side, if it's a purine, you're going to have a pyrimidine on the other side. And if this is a pyrimidine here, you're going to have a purine over there. And that's the reason that you get the base pairing between these bases and these, okay? Because one of these is a purine and one of these is a pyrimidine. And so all of that adds up to the full structure of the double helix for DNA. You've got the 5'3" direction here, you've got the cyclic form of the pentose sugar. That phosphate group there is sometimes called a phosphate ester. If you've done much organic chemistry, you'll know that this kind of pattern here, where you've got an oxygen, a central atom, and this double bonded oxygen here is a kind of ester group. And this ester group here is bridging between the 3 position of the pentose and the 5 position of the pentose all the way down the chain. So that's a repeating pattern. And then the differentiation in the pattern is according to the different bases that you have on this carbon number 1 here. And those bases have complementary base pairing to a strand which is anti-parallel. And the space between those 2 strands is sufficient to accommodate a pyrimidine and a purine. And that's what gives rise to the complementary base pairing between the adenosine and the thiamine and the guanine and the cytosine. So that's a little bit of a deep dive into this polynucleotide, better known by you probably as a nucleic acid. A polynucleotide is a polymer of nucleotides, and each nucleotide is 1 pentose sugar, 1 phosphate, and 1 of the bases. Okay, so just a short answer to all of that. So that was a long answer, wasn't it? So, but you have to choose then which of these inorganic ions here most naturally fits into the structure of a polynucleotide? Well, I mean, that one does, so you've got an answer G there, but I mean, the phosphate is actually in this backbone here as this sort of phosphate ester linkage between the 3 and the 5 rather than the free phosphate ion. But that's the phosphate ion is being used to build up the individual nucleotide before the nucleotides are polymerized into the polynucleotides. Okay, so that's, let me clean up that space down there before we get on to the next one. Okay, just clean that up a bit. There's probably a faster way of doing that. Okay, good. All right. Okay, next one is talking about an ion that's produced by the enzymic activity in red blood cells. Okay, so what you've got to think about here is that, where's my pen gone? Okay, so red blood cells have this kind of, it's sometimes described as a dumbbell shape. You have to excuse me, I haven't drawn it very well there. Okay, and these red blood cells are packed full, aren't they, of hemoglobin. And so when these red blood cells are flowing through a capillary, and there's some respiring cell over here, this respiring cell is producing carbon dioxide. That carbon dioxide is going to diffuse into the red blood cell, and within that red blood cell, that carbon dioxide is then going to react with water, and you're going to be making hydrogen carbonate anions and hydrogen ions. Okay. Now, like most biological reactions, this is going to be catalyzed. So there's an enzyme there, carbonic anhydrase, which is catalyzing this reaction of carbon dioxide and water to give hydrogen carbonate and hydrogen ions. And then because those hydrogen ions are being produced in here, because this is oxygenated hemoglobin here, you've got, I'll just do one hemoglobin subunit with its oxygen attached. And in the presence of an increased concentration of hydrogen ions, then that becomes it's called hemoglobinic acid, and the change in the hemoglobin molecule there facilitates the release of the oxygen, and that's going to then diffuse into the cell there. So that's the process that's happening close to the respiring cells. And so your answer up here, the ion that's produced by an enzyme inside red blood cells, well, the enzyme is this carbonic anhydrase, and you've got a choice, haven't you? You could say the hydrogen carbonate ion, or you could say the hydrogen ion. You've got a choice there. I think they're both equally valid answers. It's interesting if you pick up the hydrogen ion there. That's important because the hydrogen ion then binds to the oxyhemoglobin here, which facilitates the release of the oxygen molecule into the cell for respiration. So you've got a choice there, haven't you? You could either have the hydrogen ion B, that's a good choice, or you can talk about the hydrogen carbonate ion C. You've got a choice there, one of those two. Okay, good. Next one then. So the ion that's used in the production of all amino acids. Okay, all amino acids. So we just need to clean up and talk about that a little bit. Well, the amino acid synthesis needs a source of nitrogen. Now, nitrogen in the air, of course, is only there as a nitrogen molecule, which is unavailable. And so for amino acid synthesis, the source of nitrogen is not directly the nitrogen in the air, but the ammonium ion, or the ammonium ion. So there's got to be a way then to get this ammonium ion available to the plant so that amino acid synthesis has access to a source of nitrogen. So that ammonium ion there, well, the bigger picture of that, of course, is the nitrogen cycle. Okay, so nitrogen is in the air. Most of the greatest percentage of different gases in the air, nitrogen is about 78%. And to get the nitrogen from the air, the initial step for that is called fixing of nitrogen. And there's certain bacteria that amazingly can take the triply bonded nitrogen that's in the air and reduce it to the ammonium ion. And so then that could be then made available by being absorbed through the roots of plants and become available for amino acid synthesis. Much more familiar is the further part of the nitrogen cycle, where that ammonium ion is converted to, first of all, NO2 minus, and that's the nitrite ion. And then there's other bacteria in the soil that can convert from the nitrite ion to the nitrate ion. And this is the ion that's most familiar with being absorbed in the roots of the plant. But because you can see that the amino acid synthesis requires the ammonium ion, even though plants absorb nitrate within the plant, the nitrate has then got to be reduced back from the nitrate to the nitrite back to the ammonium ion before the plant can use it in amino acid synthesis. So that even though plants are absorbing nitrate, they have to do some steps first to reduce the nitrate back to the nitrite, back to the ammonium ion, before the ammonium ion can be used in amino acid synthesis. And so the other part that you are familiar with as well from the nitrogen cycle is there are denitrifying bacteria, which then return this fixed nitrogen that's in the soil back to atmospheric nitrogen. But this is the very important process through which nitrogen is made available to plants for amino acid synthesis. And if it wasn't for these nitrogen fixing bacteria over here that amazingly can work with this inert nitrogen molecule and reduce it to the ammonium ion, then there would be no starting point for the amino acid synthesis in plants. Okay, so looking back up to here then, well, the ion that's used in the production of amino acids, well, really, the starting point for amino acid synthesis is the ammonium ion, and I don't think that's in there, is it? So what they really mean is that this nitrate ion is absorbed by the roots of the plants and then is converted back to the nitrite and then reduced to the ammonium ion, and that is the starting point for amino acid synthesis. So actually that choice there, F, is the one that goes in here. Although it's a bit awkward to say that's used as a starting point for amino acid synthesis, once it's reduced to the ammonium ion, that's the one that's being used for the starting point for amino acid synthesis. This one, was the hydrogen ion, so that was answer B. And then I just wanted to talk about the heme group in hemoglobin in a bit more depth as well while we're here, because in the heme group, you've got the iron 2+ ion, so that's answer D in there, isn't it? Let's clean that up a bit. All right, so you know that hemoglobin is actually a globular protein made of 4 subunits. They're called alpha and beta, and you've got 2 alpha subunits, 2 beta subunits. And so the whole hemoglobin has this quaternary structure where there are protein chains held together. There's 4 different protein chains, 4 subunits. So that's an example of quaternary structure. So it's better to say that each of these protein chains, there's 4 of them, each of them has a heme group. So there's 4 protein subunits in hemoglobin, 4 heme groups, 2 heme groups in there, 2 heme groups in there. And the heme group isn't built up from amino acids. So the vocabulary for that is that the heme group is a prosthetic group within one of these protein subunits. It's not built from amino acids. It's called a porphyrin ring. I'm not going to draw the whole ring system here, but at the center of the ring is the iron, and then this porphyrin ring that surrounds the iron has 4 nitrogens. Now, there's a whole lot of sort of rest of the structure of that porphyrin ring, but it means that that iron in the middle there is held in place by these nitrogen atoms, which are part of a larger organic molecule, the porphyrin ring. And if you do A-level chemistry, then this is an example of ligands binding to a central transition metal ion. And in chemistry, because these look like, well, to some people, they look like the nitrogens are kind of binding onto the iron, that's called tetra, because there's 4 of them. And then this word dentate. Now that's the same word as same root as the word dentist. These are like teeth, if you like, binding or chewing on or trapping on the iron in the middle. So this is a tetradentate ligand that's bound the iron at the center of this heme group, this porphyrin ring where the iron is. Now that's looking down from the top. If you look from the side, then you've got this kind of ring system around here with the nitrogens here. And then because this ring system is part of this prosthetic group in a large protein chain, there's the sort of protein chain around here, one of these alpha or beta subunits, and there's a amino acid here which has an amine, some kind of amine group. Don't think it's NH2, it's nitrogen there, and that acts as another ligand to hold the iron in position. So the ones that go around the shape like that are axial. Sorry, my mistake. They're equatorial. And this one coming up underneath is axial. Okay. So the iron is now really held in position here, and it's only waiting, isn't it, for some kind of diatomic molecule to come on and bind in from the top. Now, in the work we've been talking about, we've been thinking of oxygen as that diatomic molecule, but you know, don't you, that carbon monoxide can be a competitor to oxygen and also bind in at the top there as well. And what happens is that this iron here is actually not in the plane of that ring. And so what happens is that when the oxygen binds to it, what happens is that this iron moves down a bit like this. And this amino acid side chain up here moves up a bit. And the combination of the iron moving down and the amino acid side chain moving up then changes the shape of the hemoglobin subunits. So there's you'd say there's conformational change. And so when the first oxygen binds, then the whole subunit itself changes shape because the iron has moved down and this side chain has moved up. And because this subunit is then connected through hydrogen bonding to these other subunits also change their shape. And it's that trigger of this kind of cascade of conformational changes that mean that when the first oxygen binds, then the affinity for oxygen at other heme groups increases. So the binding of one oxygen molecule then causes this change in the shape of the subunit, which changes the shapes of the other subunits, and then they can bind oxygen more strongly. So that's the iron at the center of the heme group, which is a prosthetic group in one of the four, well, there's one prosthetic heme group in each of those hemoglobin subunits, four of them making up the hemoglobin complex. And so you just had to put here, didn't you, that you had the iron in there, and that's a bit of a deep dive into where that iron is located and how its position as this tetradentate ligand induces this conformational change that then improves the oxygen affinity of the other heme groups elsewhere in the hemoglobin molecule. Okay, so a bit of a deep dive then into those inorganic ions there. And then we were talking, weren't we, about the nitrogen cycle and the importance of having the ammonium ion present as a starting point for amino acid synthesis. And so this next question down here is really following on from that. And you can see here, this is the process of nitrogen fixation with atmospheric nitrogen, and that atmospheric nitrogen is ending up as this ammonium ion. And this is a really incredible piece of biochemistry because the atmospheric nitrogen has this triple bond. And under almost all other circumstances, this is an incredibly inert, unreactive gas because of that strong nitrogen-nitrogen triple bond there. But in nitrogen-fixing bacteria, they're able at ambient temperatures to then fix the nitrogen as this ammonium molecule here. And your task is to try and talk all that through. So, and this is one of these sort of longer questions. You've got to talk about the enzymic behavior of this nitrogenase enzyme. So to start off with, you're very familiar, aren't you, with talking about how an enzyme binds a substrate to form the enzyme, is that right? Enzyme substrate complex, and then that goes to the regenerated enzyme and the product. So you have an induced fit or a lock and key model for that. But what the examiner wanted to see here was that not that you were talking about just an enzyme with with a substrate, but this particular enzyme has got to bind two substrates, hasn't it? It's got to bind the nitrogen gas and the hydrogen ions. And so when you're going through your description here, you're talking about an enzyme bringing together in close proximity these two substrates. You should be talking about the catalytic behavior of an enzyme. So you should be saying that to go from the nitrogen, N2, and hydrogen ion to go to the ammonium ion, you've got to get over this energy barrier here, and that energy barrier is called the activation energy. Symbol for that is EA, so that's the activation energy. And what's happening inside the enzyme is that you're providing an alternative pathway of a much lower activation energy. So that description of having an activation energy and the enzyme giving you a lower alternative pathway for a smaller activation energy is something you want to have in your description as well. And I think really at this point, instead of just using a lock and key model, it's better to have an induced fit model, because when these two substrates are bound in the active site of the enzyme, that changes the shape of the enzyme. So there's a conformational change in the shape of the enzyme. And it's that conformational change in the shape of the enzyme that is putting strain on this nitrogen-nitrogen triple bond. And that means that that can then be reduced in this process here in a way that's easier than trying to go through the high activation energy barrier there. So you're talking about induced fit, you're talking about the conformational or the shape change of the enzyme, and that's putting strain into this nitrogen molecule, which is then allowing it to be reduced from the nitrogen molecule to the ammonium ion. You can see here that that's a reduction, and in any reduction is a gain of electrons, and so those electrons have got to be supplied from somewhere else. And if we were using sort of A-level chemistry language, then you would say an oxidizing agent is providing the electrons. And the other point the examiner wants you to put in here is that that process, you can see you're breaking a triple bond there. That's going to be consuming ATP. So let me just clean that up. So ATP is being consumed during that process. And actually there's 2 molecules of ATP used for each of these electrons here. So you end up with 16 molecules of ATP, going down to 16 molecules of ADP. So I think that covers most of what the examiner was looking for there. There was another point that at the active site of these enzymes, there are these transition metal ions. Okay? And those can very easily be oxidized. And that would interfere with the catalytic behavior of the enzyme. And so this enzyme is operating under conditions where there is no oxygen, which would essentially poison the catalytic activity based on the vanadium and the molybdenum ions in the active site of the enzyme. So this is, you've got to say this is happening under no oxygen, this is happening under anaerobic conditions. Because if there was any oxygen present, then the vanadium ion and molybdenum ions would be oxidized, and they wouldn't be available to work in the active site of this enzyme. Good. Okay, so this whole question is about inorganic ions, isn't it? Because there's another part of the question down here. So this is looking at these ions here, and the soil concentration or the surrounding solution concentration is low. You've got a concentration here in millimoles per decimeter cubed, but the concentration within the root tissue is much higher. And so these ions are being moved from an environment of low concentration to an environment of high concentration. So that means they are being moved into the root cells against a concentration gradient. Now, with these kinds of questions where they say you've got a reference to table 2.2, then a really good thing to do is to actually reference the data that's in the table. So we'll come back to that later on, but in your explanation here, you've got 5 marks to get. You should be making sure that you've actually used data from this table as part of your explanation. Okay, so you've got to start, I think, by describing the phospholipid bilayer for the cell membrane. So if you remember, you've got these phospholipids which have a hydrophobic tail and a hydrophilic head, and you end up with this kind of arrangement. So that's the head there, and that's the hydrophobic tail. That's a hydrophilic head, and that's the hydrophobic tail, that bit there. And if we just blow those up a bit, you've got this glycerol molecule, and 2 of those alcohol groups on the glycerol molecule are esterified, and you have these fatty acids here. That's where the long chain is, and there's another one there with this long chain. That's the hydrophobic part, and the hydrophilic part is because this alcohol group here from the glycerol is bound to a phosphate group. So this is ionic, that's the hydrophilic end. I'll just say it. That's the hydrophilic end, and then these long fatty acids, carbon chains here, that's the hydrophobic end. And so they align themselves like this to give a bilayer, and it means that this middle part here is all hydrophobic. That's where the fatty acids are. And because these ions up here are all charged, then they can't move through this hydrophobic part in the middle here. So the ions are stuck out on the outside there, and on the inside of the cell, once they're through, they can't diffuse back because of this hydrophobic interior part of the phospholipid bilayer. And so the only way to get them from the outside into the inside is to have a protein which spans the membrane, and is going to provide a kind of channel or pump to allow these ions to get through. And because that's an example of active transport, because we're moving these ions against a concentration gradient, then there's going to be energy required to make that process happen, and you're going to be saying that ATP is consumed in the active transport of the ions from the low concentration on the outside of the cell to the high concentration on the inside of the cell. So that's the sort of biochemical basis of the sense of this question. But in terms of getting the marks on this, then you've got to reference some of the data in this question. Now you can see here that if you take potassium, then the concentration gradient is particularly high. You've got a 75 millimolar concentration on the inside of the cell. You've got a 1 millimolar concentration on the outside of the cell. That's a particularly strong concentration gradient. And if you look at this one here, that again is a particularly strong concentration gradient. And then for some of these other ones, like for the calcium one, you've got quite a shallow concentration gradient, going from 1 millimolar to 2 millimolar. You're still going against the concentration gradient, but it's quite small. So you can start to pick up some of the data from the table here to talk about which ones have a stronger or a greater concentration gradient. And if there's a greater concentration gradient here, that's indicative that there must be more potassium channels, potassium pumps, or sulfate pumps than there are calcium pumps, or calcium channels. So just in terms of the proteins that are crossing the cell membrane here, there's going to be more of the ones that do the potassium and the sulfate transport than there are of the calcium transport. Even if that's not something that you should know, you can deduce it from the table here, and that use of data in the answer is one of the things where you can show that you've referenced the data in table 2.2. So I think we've covered most of the points there. Oh, the other thing you should point out as well is that because these these proteins that span the cell membrane are using active transport, they are specific for each of these different ions up here. And that's another reason why you're getting the potassium and the sulfate moving against a particularly large concentration gradient, is because there's specific pumps for potassium and sulfate, and they're different to the specific pump for calcium. So, talking about the specificity of the pumps and the active transport is something that you can pick up from that data table as well. And then you can see here that the experiment was run, wasn't it, over 3 days? And that means that as this active transport process happened, and these ions were pumped from the surrounding solution into the root tissue, there's no way that that process could go backwards. They can't passively diffuse out. Why not? Because the middle of this phospholipid bilayer, the cell membrane, has a hydrophobic center to it, and that stops these positively charged ions moving back out. So once they're through the active transport, if you like, they're trapped within the cell to be used, and they can't diffuse out again. Good, so that was question 2 there. A lot of work really on these inorganic ions. Okay, the other one I wanted to look at in a bit of depth was question number 5. Okay, so you've got here DNA triplets. So these are part of the genetic code, and we've got a correspondence here between the triplets in the genetic code and the amino acids that end up building a protein. And so this is a genetic dictionary for DNA. Okay. You would have a different genetic dictionary for messenger RNA and a different genetic dictionary for transport RNA. And this here is the genetic dictionary for DNA. And you've got to talk through in this question the process of transcription, where the genetic code triplet is transcribed to a messenger RNA codon. And then you've got to talk about the process of translation, where the anticodon of transport RNA is used to bring a particular amino acid to a ribosome to build up a protein chain. So you're given that genetic dictionary there, and then you've got to work out some missing parts here. So you've got to know that the complementary base pairing is adenosine and thymine, and you've got to know the complementary base pairing is a cytosine and guanine, and that's the complementary base pairing in DNA. And the complementary base pairing in messenger RNA is, you've still got, so that's 3 hydrogen bonds there, it's still that, isn't it? But in messenger RNA, you've got complementary base pairing between adenosine and uracil. Okay, so there's that difference between the RNAs and the DNA. So you've got a case of transcription here. Well, if you've got that, these are triplets in the DNA genetic code here, and these are codons, aren't they? And so if you've got complementary base pairing here, then the C must be giving you a G there, the A must be giving you a T there, and the C must be giving you a G there. So you can work backwards from the codon in the messenger RNA to the triplet that must have coded for that in the DNA. This is a little bit more straightforward. You're now going from a triplet to the codon, and so the T is going to have a complementary base pair for adenosine there. The G is going to have a complementary base pair of cytosine, and the A, it's this one here, isn't it? So they're just checking that you're making the right complementary base pair for messenger RNA. So that's going to be A, C, and U. Okay, and then because we had a genetic dictionary for the genetic code DNA, then when you come to looking at the amino acid that eventually gets built into the protein, you're looking up in the genetic dictionary not for the messenger RNA, we didn't have that, but the genetic dictionary for the triplet code in the DNA. So we've got to look up and see what is the amino acid that's being coded for by that triplet. So it's that one there, isn't it? So that amino acid, leucine. Okay. All right. So, well, and then I suppose we're being prompted a little bit here to think about protein synthesis from individual amino acids. Well, the sequence of amino acids in protein, polypeptide, of course, is primary structure. That this structure here, this sequence of the amino acids, is the primary structure in the protein. Now, we've got a sort of longer question here. Again, you see there's these 5-mark questions. You're actually being, you've got quite long answers to give here. And they're saying that if you look at the sequence of triplets here, that's the first, second, third, fourth, fifth, sixth, seventh triplets. There's a single point mutation here, where one of the bases here is not the base which leads to normal hemoglobin. So one of those bases is going to give valine and not that amino acid there. So in sickle cell anemia, the hemoglobin has an amino acid of valine at that point rather than glutamic acid. So we have to look up in the genetic dictionary then. So for valine, we've got to find something up here which has a single point mutation. So we should have in an unmutated DNA CTC, and if we've got a single point mutation for valine, that's going to give us, where's valine? That one. So instead of having CTC, we're having CAC. We've made just one base is wrong. We've got adenosine instead of thymine. And in the genetic dictionary, that's sufficient to code for a different codon there, which then is going to give us valine in the protein later on. So I think in this answer here, you've got to first talk about transcription. And you've got this single point mutation from the normal gene to the mutated gene, which is CAC. We saw that just now. And so that sixth triplet, CAC, now is then going to become GUG, isn't it? So that's the triplet, and this is going to be the codon in messenger RNA. So that's GUG. Okay, so then the messenger RNA is then going to be moving to a ribosome. And you then have particular transfer RNAs with one transfer RNA for each amino acid. So the transfer RNAs have this kind of structure, where you've got that kind of shape, and the particular amino acid is attached at that point. So in this case, for us, it's valine, which is being added to the protein chain, and then there's going to be an anticodon here, which is complementary to the codon that was transcribed, GUG, from that point mutation in the genetic code, CAC. And so the codon there is going to be CAC as well. So you've got to say that that the messenger RNA is responsible for the order of binding, because there's a sequence to the messenger RNA, and so that's going to be a complementary base pair, that codon there to the anticodon there, and there are specific transfer RNAs, so one transfer RNA for each of the different amino acids. And so what's going to happen is that the messenger RNA will form the will specify the sequence, and then the transfer RNA will bring the valine amino acid, and that'll then become the next amino acid to be added to the protein chain that's being built. So in that question there, you've got to work on transcription, and then you've got to talk about translation as well. The transcription is the converting of the genetic code into the codons in the messenger RNA, and the translation is the use of each of these different transfer RNAs with their particular amino acids. And with the genetic code, you have a triplet. With the messenger RNA, you have a codon. With the transfer RNA, you've got an anticodon. Okay, so those were the questions I wanted to focus on in this masterclass. And we've been particularly focusing on biochemistry, looking at all these different inorganic ions that we had up in question 2, and then looking at the process of using the genetic code to transcribe for the codons in messenger RNA, and then the messenger RNA codons being having complementary base pairing to the anticodons in transfer RNA, giving rise to the sequence of amino acids in the protein chains that are built in the ribosomes. Okay, very good. So that's as far as we're going to go with this paper 2. If you want to follow up with the Z coaches, then the Z coaches are on the Slack channel from Cambridge Leadership College. And it just remains for me to wish you every success in your studies of biology, and look forward to joining the next masterclass from Cambridge Leadership College.